Oh.
Sir, good morning, sir. Just we are waiting for our correspondent principal, sir. Please be online, sir.
Sure, madam.
Okay, sir.
Hello.
Good morning, madam. Solluங்க madam.
Sir, good morning, sir. Principal sir told that he will be coming, sir. So shall we start?
Correspondent sir.
Among, sir.
சரி நீங்க ஆரம்பிச்சிருங்க. அவர் என்ன சொன்னார், ஜாயின் பண்ணிக்கிறேன்னாரா?
ஆமாங்க சார்.
சரி நீங்க ஆரம்பிச்சிருங்க. ஏன்னா 10 மணிக்கு அவங்க ரிசோர்ஸ் பர்சன்ஸ் ரெடியா இருப்பாங்களா?
ரெடியா இருக்காங்க சார்.
சரிங்க ஆரம்பிச்சிருங்க.
ஓகே சார்.
My pleasant morning greetings to all the dignitaries, faculty members, research scholars, and students to the one-day national virtual seminar on emerging technologies in energy devices and functional biomaterials on the behalf of Department of Physics, Unga Arts and Science College, Erode. In the age of information, ignorance is a choice, action is a foundational key to all success, and it requires effective participation. I wholeheartedly welcome all the participants of the convention. It's a privilege to share that our department is one of the departments among 5 departments recognized in the DBT Star College scheme and funded with the grant of 1.04 crores by the Department of Biotechnology, Government of India. I extend my invitation to the gathering to invoke Lord Almighty to illuminate the sparkle of knowledge among us. Prayer.
கலை நிறை கணபதி சரணம் சரணம் கஜமுக குணனிடே சரணம் சரணம் தலைவனின் இணையடி சரணம் சரணம் சரவண பவகுக சரணம் சரணம் சிலை மலை உடையவா சரணம் சரணம் சிவ 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 சரணம் சரணம் உழைவர் மோரு பாறை சரணம் சரணம் உமை சிவயம்பிகை சரணம் 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 சரணம்.
Welcoming is a new beginning with endless possibilities. I invite our head of the department, Ms. R. Chitram, to welcome the gathering. Please, ma'am.
A very pleasant morning to all the distinguished delegates and participants. It is a glorious moment to extend my warm wishes to all on behalf of Pung Arts and Science College, Erode. It gives me tremendous pleasure and honor to welcome you all for one-day national virtual seminar on emerging technologies in energy devices and functional biomaterials. On behalf of Department of Physics, I wholeheartedly welcome our respected correspondent sir, Thiru A.K. Palanisamy avargal, who has always been a supporting pillar in all our activities. I am delighted to offer a hearty welcome to our beloved principal, Dr. N. Raman sir, who is earnest guide to all our endeavors. I am highly honored to take this opportunity to welcome our resource persons, Dr. Sarat Chandra Virla, Associate Professor, Department of Humanities and Basic Sciences, Godavari Institute of Engineering and Technology, Rajamundry, Andhra Pradesh, and Dr. T. Elumalai, Professor and Head, Department of Green Energy Technology, Madanji School of Technologies, Pondicherry University, Pondicherry, for accepting our invitation to share their knowledge in the field of trending research on biomaterials and energy devices. I extend my heartfelt welcome to all the faculty members, research scholars, and students from all over India who sincerely committed to this event to make this success. I assure that the seminar will be highly engaging and beneficial for your future ventures. I welcome you all once again to the seminar and hope that you will have a great time ahead. Thank you all.
Thank you, ma'am. Leaders don't create great followers. They create more leaders. I thank our respected correspondent, sir, Thiru A. Palanisamy avargal, who always guides us to empower. I thank sir's support, and with his wishes and blessings, we would like to proceed further. Coming together is the beginning, keeping together is progress, working together is success. I welcome our beloved principal, Dr. N. Raman sir, who leads us to profound path of success to felicitate the gathering. Please, sir.
Good morning to one and all present here. At the outset, I extend a cordial welcome to all the resource persons, faculty members, and participants for this one-day national level seminar. First of all, I congratulate Professor Chitra and her team for organizing this national level seminar. It's the first time of the seminar organized by the Department of Physics, right from the establishment of its program in our college. The topic which was selected by the team also is novel, and as well as it is apt for the current moment. I hope the participants get in a kind of information from the resource persons, and it will be very much useful for them for their career also. With these few words, I wish the program as a success. I once again extend a cordial welcome to all the resource persons and the participants for this seminar. Thank you for the opportunities given to me.
Thank you, sir. I request Ms. V. Vishali, Assistant Professor, Department of Physics, to introduce our chief guest of the session, Dr. Sarat Chandra Virla, Associate Professor, Department of Humanities and Basic Sciences, Godavari Institute of Engineering and Technology, Rajamundry, Andhra Pradesh. Please, ma'am.
A warm good morning to everyone connected. I take great pleasure to introduce our renowned resource person of this session, Dr. Sarat Chandra Virla, Associate Professor, Department of Humanities and Basic Sciences, Godavari Institute of Engineering and Technology, Andhra Pradesh. The core research areas of our honored guest are biomaterials, synthesis of nanoparticles for hyperthermia treatment and its drug delivery, A warm good morning to everyone connected. I take great pleasure to introduce our renowned resource person of this session, Dr. Sarachandra Veerla, Associate Professor, Department of Humanities and Basic Sciences, Godavari Institute of Engineering and Technology, Andhra Pradesh. The core research areas of our honored guest are biomaterials, synthesis of nanoparticles for hyperthermia treatment and its drug delivery, and defluoridation from groundwater using different sorbents. The research journey of our guest of honor began from his doctorate at Anna University, Chennai, Tamil Nadu. During this period, he received CSIR Senior Research Fellowship and National Doctoral Fellowship from AICTE. Dr. Sarat Sir received Erasmus Mandus Postdoctoral Fellowship during the period 2013 to 2014 and sat at his postdoctoral fellowship at Politecnico di Torino, Italy. Our guest of honor was a research associate in the Department of Aerospace Engineering, Indian Institute of Science, Bangalore. Dr. Sarat Sir was research professor at Chosun University and Chagnam National University, South Korea. Our resource person has published 26 research papers in various international reputed peer-reviewed journals and book chapters in Handbook of Polymer and Ceramic Nanotechnology. Furthermore, SAR has filed a patent titled Medium for the Removal of Fluoride from Water. Our revered chief guest has been acting as reviewer in peer-reviewed international journals, Journal of Allies and Compounds, Elsevier, Archives of Oral Biology, Elsevier, Material Science and Engineering, Elsevier, Journal of Biomaterial Science, Polymer Edition, Taylor and Francis. Besides, he is a lifetime member in Society of Biomaterials and Artificial Organs. It is our fortune to have such an inspired person in this session. Thank you.
Thank you, ma'am. It's a teacher that makes a difference and not the classroom. I welcome our resource person who created difference in lives of many students and research scholars. I wish all the participants to gain the insight and enhance the level of understanding in the arena of functional biomaterials as a life-saving drug. Now I request the resource person to take over the session. Please, sir.
Thank you, madam. First of all, I would like to thank correspondent sir of Congo Arts and Science College and our beloved principal Raman sir and Department of HOD, Department of Physics, Congo Science, Arts and Science College. So Chitra ma'am. So thank you for giving me the opportunity to deliver my lecture on my research topic. All right. So coming to my research area, so as I had vast experiences of past 1 decade, I'm doing my research on biomaterials, like fabrication of biomaterials for the usage of different various fields like defluoridation. Defluoridation means like so fluorine absorbent content, like so whatever I use my synthesized biomaterial, so which we can keep in the material of a groundwater, whatever the groundwater, which can be absorbed of fluorine content up to the 92 percentage. So that is about my some of the research areas which I worked. And coming to me, I'm Dr. Sharat Chandra, which I did my PhD at Anna University and all those things like. So currently I was working at Godavari Institute of Technology. We established a new lab, which is called as Nanomaterials for Photovoltaic and Biomaterials Laboratory. So which we had received a seeded fund from our institution. And then with that, we had applied different various projects also at the DBT as well as the DST. So soon we are waiting for the positive response from the government agencies. So coming to this, this is about my introductions and coming to my area of research. So these are all my research areas which I had used in the. So these are all my research areas like controlled nano-sized bioceramics for the biomedical applications. So as the biomaterials are bioceramic materials which could be used for the different area like heart valves and also like bone and dental replacement materials also. So as we had like teeth also, we have dental for implanting those teeth materials also, these bioceramics. Mostly these bioceramics which I had used calcium phosphate. Calcium phosphate is one of such materials, so which could be used as a better biocompatible material. So what is biocompatible? Like biocompatible, even if we include implant inside our body also, so that could implant mimic our body inside the human body. So and coming to the different diseases like bone diseases, which I had shown, like osteosarcoma. Osteosarcoma is nothing but like bone cancer treatment, bone cancer. So and osteoporosis. Osteoporosis is due to the deficiency of the calcium phosphate. So that's what this osteoporosis mainly can be occurred in the old aged people. Like since they have, so for the old age people they have so osteoporosis and all. So that's what they will intake those calcium tablets and all those things. And coming to the skeletal fluorosis and dental fluorosis, which is the excess of fluorine content which we take so through the drinking water and all. So this skeletal fluorosis and dental fluorosis will occur. Like so, some other diseases like osteonecrosis, and mostly this will be happen for the childhood children's so when we excess of eating sweets and all those things like chocolates and all those things like osteoarthritis. Osteoarthritis is nothing but like the excess of bone growth inside our body and all. So that will be called as osteoarthritis. Like osteomyelitis is like the bacteria, some of the bacteria which will be having so E. coli, staphylococcus, and all. Due to that reason, bacteria. So these osteomyelitis will occur. Like some of those examples, like applications which can be used for the bioceramics, which is nothing but like a so targeted drug. The beauty of these targeted drug delivery, which can cure the osteosarcoma disease. And my research area, which I had used, implant these bioceramics with an metal ions, so that these metal ions plays a major role to act as a hypothermia treatment. What is hypothermia? Is nothing but normally our body temperature is up to 37 degrees Celsius. So about that, so due to this metal ions incorporation in the ceramic material, bioceramic material of these metal doped bioceramics, which acts as an so hypothermia, and so that it can cure the cancer cells. It can kill the cancer cells by without damaging the normal cells. And coming to this synthesizing, I had obtained with different various technology techniques, so which is like hypothermia, sol gel, and gel techniques by synthesize these bioceramic material, and which could be used for our daily life, like biomedical applications. So as the beauty which I had found was like this bioceramics normally usually is synthesized in the hydrothermal. Hydrothermal is nothing but like I will discuss in the next few slides, like whatever the all the so spheres initially which I had obtained nanosphere morphology. So nano-sized morphology of the spherical shape of bioceramic calcium phosphate material. And slowly when we incorporate these metal ions into this ceramic material, which could be changes towards the spherical to the rod-like morphology kind of things. And after that, so whether this biomaterial, whatever I had synthesized, those biomaterial, whether my synthesized material that could be used in the bio applications or not, we need to check with the different organisms or like a cell culture and drug delivery, blood compatible. We have to obtain all the various techniques like analysis which we have to do. Then only we can finalize this biomaterial could be used in the human body. Unless until without doing those physical characterization and simply if we can say this will be useful for the biomedical application, that won't work out. So mostly, so nowadays the publications which we will be incorporating with the mostly with the in vivo studies also. In vivo in the sense like, so what in vivo means, like mostly this synthesized material which we can implant into the mouse, rabbit, or some other material. So living organisms, like living things. beings, like which could be, if it's sustained, even if we implant those biomaterials, then we can say that will be the better biocompatible material. So slowly, what we had studied is like, as my material which I said is a magnetic metal doped. Like metal is nothing but like an iron, cobalt, and nickel, which shows the better magnetic property. Like, so due to this magnetic property, so this will shows very good nature, very good property of so human cancer treatment and tumor treatments also, like. So after that, like drug delivery also, which we had published with the recent publications on drug delivery applications also. And also which we had worked at our laboratory at Godavari Institute of Technology. So what we had also doing with different piezoelectric, one of my colleague, which is Dr. Ashtakal Anil Kumar, which we are collaboratively working on this piezoelectric materials, which could be used for the energy harvesting material. Like, so what would be the energy harvesting is nothing but like even if we have the cars and all, some of those videos which we can have, so those representing that, like even if we have show the cars and similar material like lorries and all on the roads, those due to the stress and stress on that material on that road, like electrodes, so that can generate the electricity. Like, so due to that reason, which we are also obtaining with, uh, so you can see this some of those our material which we had synthesized. So those electrode by spin coating method. So after that, which we are showing that electric nano generators, which could be used as a electric nano generators. Like, so mostly this will be occur. So due to the piezoelectric effect and all right. So coming to this, like what is nanomaterial and all those things, like so which says that like nanomaterial is commonly so I'll discuss about the similar kind of things with just a minute, like I'll show you so coming to this, like as I would like to discuss deeply with the students also, so which would be beneficial for them. So since from the basics and all, like I would like to overall those session with the, so what is nanomaterials kind of things, like I'll discuss all deeply with those things, like so that it would be useful to understand, better understanding for the students and all, like. So nanomaterials, simply just like nanomaterials are commonly defined as the materials which will be the average grain size, that would be the 100 less than 100 nanometers. Like, so what we had synthesized those metal doped iron oxide, iron doped material. So which says that like which iron doped, so which we synthesize, which it has obtained till up to 20 nanometer range. So those 20 nanometers and all, which we can observe through the transmission electron microscopy and all those things, like. So as the paper sheet, which is about the thickness of 10,000 nanometer thickness, and DNA is having the 2.5 nanometer range and all. Even I think like some of the slide in the next few slides, I had added the corona, the size of those corona virus also, which I had, which has been focused through the transmission electron microscopy and all kind of things, like. So hope you like the human DNA is 2.5, and even the human hair and a single gold atom which is third of the nanometer diameter. Even gold nanoparticles which can be observed. So some of the papers which has been observed to up to the 5 nanometer range also, like. So these are all the different ranges which I had discussed to better understanding for those things, like. And coming to this nanotechnology could be used in various vast fields, like which is nothing but like optical engineering, biomedical, cosmetic, textile, and also metallurgy, and even for the defense also. Defense mostly they are using like this nanomaterial, nano coatings. They are used for the aircrafts and aeroplanes and all. Mostly these nano coatings and all, like so that it can withstand very high temperatures also, like. So coming to this, like, so as we have a nanomaterial, so with an ultimate technology, like, so as we know that most of the lizards and some other things which can be, even they can walk on the mirrors also, like, so how could it be like, so there will be a gel which is nothing but like a nano gel, dry adhesive material which has been sticked on the bottom of the hands and legs, all kind of things for the lizards and all that. So those things which can also be observed, even the, so recently it has been published like in the nano letters, like so GECO principle, which can be said that as an a GECO principle, like so those adhesiveness which has been sticked on the hands and palms of those materials, due to that adhesiveness, so that adhesiveness which will be also in the nanometer range, like so due to that adhesive property, so this has been sticked on the walls, and as well, even if the weight of the body is very high, so that can be stick very high adhesive property which has been on the hands and legs. So that's what it can run away on the mirrors and even on the walls of the things, like. Coming to the classification of biomaterials, like even in the nanomaterials also, which you can say that, like, so whatever these biomaterials like graphene, carbon nanotubes, all these graphene and all, graphene has the beauty which has been used in the drug delivery applications, like mostly they are all using these graphene material, so which we had already published in ACS applied materials and interfaces, like graphene doped so scaffold material which we had published, like so those scaffold materials which gives the a better property for the cancer, for the tumor targeting drug delivery applications and all, graphene doped scaffolds, like so this differentiation of those biomaterials, which is nothing but like uh, so which is nothing but zero core shell nanoparticles, is quantum dots, halo cubes, so which is nothing but zero dimensional quantum dots, like so which says that the zero dimensional quantum dots and all hollow tubes, which is nothing but says that like a core shell morphology kind of things, whatever those material which can be no dimension in that, like so which will be spherical in shape and all those things, like so, so coming to the one-dimensional, mostly which we can even some of those nanotubes, one-dimensional nanotubes and nanorods, which can be observed in my paper, one of my paper, so which is nothing but nanorods, nanotubes, and nanobelts, and nano ribbons, like kind of things, like. So coming to those things, like like so. So one-dimensional nano wires and nano rods and nano belts or nano ribbons, like kind of things. Coming to the two-dimensional, a nano fiber and nano plates, like two-dimensional is nothing but mostly this has been used as a scaffold materials, like. So what is scaffold materials? Which you may can ask some of those questions, like scaffold material is nothing but mostly for the wound dressing and all kind of things. When we have the patches and all those things for wound dressing and all, so they are mostly they use this scaffold kind of materials like polymer. So mostly this will be a polymer based material, biomaterial, which can be make it in the even if we have some wound on that, So if we keep those scaffold materials, it can mimic inside those scaffold skins also. So this could be used as like scaffold material for two-dimensional robotics. And coming to the three-dimensional nanoballs and nanocoils, like so mostly this mesoporous kind of structure, which could be used for the drug delivery applications kind of thing. Like so through those porous kind of things, even if we inject some of the drugs inside our body, so through this, based on this mesoporous structure, mostly for our human bone, if we break those bones inside, there will be a porous kind of structure will be obtained. So due to that porous structure only, this drug and all will can deliver from one site to another site and all. So these mesoporous structure, which could be used for the biomaterial kind of things. So as I shown this, all these zero dimension, one, two, and three dimensions. So based on that, I'll give you some of those examples of those things like through the which could be seen under the microscope, like traveling microscope, transmission electron microscopy, and even on the scanning electron microscopy. Like so, whereas zero dimensional core shell nanoparticles and quantum dots, so which could be used as like a cobalt ferrite nanohydroxyapatite core shell structure. So this was the picture which I was showing, like so core and shell structure kind of things of the cobalt ferrite. Cobalt ferrite is the best for the metal doped, like metal doped material, which could be used for the as in like targeting magnetic oxide, iron oxide, all these oxide materials which could be used as a targeted drug delivery applications. Like so, even for these cobalt ferrite material also used as a targeted drug delivery application. Like so, this nanoparticle, the cobalt ferrite nanoparticle, which shows better magnetic property. Due to that reason only, so this has been targeted the particular. So what, how we can target those structure through this metal domains? So inside that there will be a magnetic field which we can keep in that, like so where we have in the output, so wherever the magnetic field, if we can see in the in my previous slides, so those nano particles, magnetic nanoparticles, which could target at the magnetic field site. So there, these hypothermia treatment, hypothermia will start doing like, so what that hypothermia will do is, it will slowly increasing the temperature. Like, so due to this external magnetic field, so these magnetic nanoparticles will makes that with the heating with till up to 42 to 45 degrees Celsius. Like, so even if we go extend with that temperature, so which that could also kills the normal cells, which will not be useful for our material like synthesizing. Like even if we keep on increasing the temperature, our body can also getting heat, and then slowly it will there will be a several disadvantages also will occur in that. All right. So what the thing is like, so one-dimensional nano wires, nano rods, nano tubes, and nano bolts and nano ribbons. Like so, what nano ribbons is nothing but like so nano ribbons and nano rods is nothing but like iron doped nano items. So as I had my synthesized material like which we had synthesized in the hydrothermal as well as the combinatorial technique which I had used like hydrothermal as well as the microwave. So through those hydrothermal and microwave techniques, which were beautifully, which we had observed those nano rods and all kind of things like through spherical morphology to the nano rod like structure morphology. Coming to the two-dimensional, as we have like nano belts and nano plates and nano discs and all kind of things like. So as we have three dimensions, will show that the nanoballs, nanoballs and nanocoils and nanopillars kind of thing. So some of those examples which I had given, magnesium ferrite, magmite for the hypothermia, as well as the graphene nanosheets for the tissue engineering application. Like as I said, a tissue engineering application is nothing but like even if we have a scaffold material, these patches kind of how we can use the band-aid. So similarly, this graphene material nanosheets which we can patch on that wound material at the above of that wound, so that slowly the graphene and all which can be reacts easily, and so that it can cure as soon as possible for this material kind of thing. So these are all the some of the materials for the nano applications like different drug delivery sensors, bioimaging and all tendrimites, and even with the biomedicine and anti-cancer and gene therapy. Even nanoparticles, magnetic nanoparticles will have silver, gold, and platinum composite kind of things. Coming to the scale of things like just a minute, like coming to the scale region and all. So what this has been says that like even the blood cells and all those things like the things is nothing but like natural and all those things like human hair and flour and red blood cells and with white cells, and even the interferon which will be having the virus. So virus will be cured the drug which will be having 10 nanometer range and all those. So this is the things which has been natural use, as well as the things which could be used for the man-made kind of things like so coming to this like this is the even the red blood cells which will be having 2 to 5 nanometer range and all kind of things like so coming to this, detecting cancer cells, which is nothing but what is cancer cells which will be having without nanoshells and with nanoshells kind of things like. So coming to this silica nanoparticles coated with the gold and all, as I said with the core and shell nanoparticles like. So what they had done is like, so gold has been having the very good property of the cancer treatment property like. So that's what like so gold can cure the cancers of the several cancers of breast cancer and also different kind of things like. So coming to this, whatever the things, so breast cancer detecting those cancer cells and all, so which could be used as a small silver core shell kind of things like gold nanoparticles, which could be saying that like advantage enhances the detection of cancer cells in the real time. So this silver will have the very good photoluminescence property like. Am I right? So due to this photoluminescence property, what this luminescence bio tracking and all, which we can track those nanoparticles of core shell nanoparticles of silver nanoparticles at the target site, so that when it reaches at the target site, these gold and all, silver and gold and acts as an very good curing of cancer cells and all, so that it can target those cancer cells and which could be cures the cancers. And then this is the detection of the cancer cells in the real time like. So some of those examples of those without shell and with nanoshells, which we can see those silver nanoparticles. This will be the normal is saying that without cancer cells and all. So HR2, HER2, as well as the HER2 plus cancer in the sense like with cancer and without cancer, as I differentiate those cells and all. So that what we had observed is most of these cancer cells and all which can kill those cancer cells like. So coming to the synthesis of those nanoparticles and all like which it says that like top down approach as well as the bottom up approach, which says that like From the top to down means is nothing but like from bulk material to the nano material, which we can slowly what we can do is like ball milling and all these things. What we did is, so we have a very big nano micrometer sized material which we can grind those through the ball milling process and all, so that which could be making into the nano formation. Like, and coming to the bottom to up is nothing but like, so slowly these precursors like soul gel and all. So through the water as a medium or alcohol as a medium, if we keep doing those soul gel and gel technique, so from from micro nano size to which we keep on increasing those material, so that the formation of precipitation will also observe in through this gelation formation or solution formation. So that after making this powder precipitation technique, and we can grind those material thoroughly, and then we can make it into the nano size. Like, so from top to bottom we can synthesize, as well as the bottom to top also we can synthesize those material. Like, and coming to this, like top down approach is nothing but milling or attrition. So thermal cycle and 10 to 1000 broad size distribution, varied particle shape or geometry. So, so these are all the different approaches which we can synthesize those ball milling and annealing and dip in lithography and kind of things, like for nano composites and nano grain bulk sizes and all. Like, so coming to the bottom-up approaches and all, scientists' curiosity to understand, mimic how biological architecture are programmed as a self-bioassemble as well as a self-organized material. So from coming to the bottom-up approach is, so mostly due to the interaction between the pi and van der Waals interactions of the electrostatic, and so that hydrophilic and hydrophobic interactions between the molecules and molecules, so we can synthesize those in the natural process kind of things. Like, and coming to the various applications like chemical biochemical technologies and electrical and photons, as well as different types of bottom up approach. So mostly, so for currently most of them are approaching as a bottom up approaches. Like, so these are all the different synthesized techniques like liquid phase, gaseous phase, and template based and template free based kind of things. Like, so mostly template free based is nothing but like so hydrothermal, sonochemical, and surfactant assisted kind of things will comes under the liquid phase bottom up approaches. Coming to the gas phase, so mostly like coating and all, like gallium nitride and all those other techniques and all, which could be observed through the gas phase and all kind of things. Like, so coming to this, simply whatever this soul and soul gel, how it has can obtained and all, we can see those soul gel and so soul gel through this soul gel through solutions, like two solutions which we can mix together in the alcohol as a medium. Like, so we'll take alcohol as a medium and then we'll mix together whatever the chemicals which we have to synthesize, and then slowly after stirring and all, we can keep it in the stirrer, and we can making stirring up to 2 hours 3 hours, and after that we gelation formation. So through this whole alcohol and all alcohol, so these there will be a gelation has been formed. That's what this will be called as an soul and gel. Soul is nothing but solution, so as we take as a solution in the initially, and then slowly after obtaining a gelation has been obtained at the end product. So starting product we will take as a solution based. Like, so after those end product or final product which we have washed with different alcohols or water or whatever those things, and so that we will remove all those alcohol based kind of things, and then we can do the those drying and all, we can obtain those material with after drying, and we can get the fine powder and kind of thing, so that we can take those material into the under the microscope or different XRD phase conformation and all those things. So this is about the soul gel. And coming to the hydrothermal and solvothermal, like, so what is hydrothermal is nothing but like Hydro means water, like, so thermal means treatment, like, so under high pressure and high temperature, this nano sized material can also absorb in that, like. So what this is one of my paper which we had published in ACS. So what this says is like a spherical morphology which you can be observed through the transmission electron microscopy, like. So what we did is like indigenous hydrothermal, which we had done developed in the at Anna University with under my professor, PhD professor. What we do is like we have synthesized those material in the Teflon beaker at high temperatures and high pressure, like. So we will close with the enclosure, and then we will synthesize this material, and then we can beautifully obtain those bioceramic in a spherical morphology. And slowly, slowly, if we keep adding those metal ions, so what these metal ions plays a major role, and these spherical formation of nanomaterial will be slowly converting into the rod-like morphology kind of things. Like. And coming to the next one, which I had done with the photopolymerization, what is photopolymerization is nothing but like, so as we have, as I said, this is one of the biomaterial, like scaffold material we synthesize with the polymerization. So what we do is like, so PMM, polymethyl methacrylate, is one of the polymer which we had obtained, like biopolymer. So which we take under the photogram, like photo light, so UV light, like. So through those UV light and all, we this polymerization takes place. This cross linking and all we can take place in that material, and then slowly it will be mineralized, and then it will obtain as a gel kind of things, like kind of material, like it will stick to the other, like. And after gelation formation, we will keep those material into the freeze drying, and those after freeze drying, which we can upset the final product, which we can show you. So there here at as a scaffold material, and then this material which we had through the under scanning electron microscopy, which we can scaffold material slowly, like there will be a chain formation in the images at 5 nanometer range, like kind of things which we can observe. Coming to the electron biosensors and all, what I did at the Chungnam National University at South Korea, what we do is like different substrate material, like glass as a substrate material, silica glass, which is substrate which we can keep, and then slowly which we do the bio coating, polymer coating in that on the material as a polymer coating through by using spin coating method, like. And after that, which we have as a used as a shell, it will be under process, which we are going to publish soon. In that, I don't want to reveal those much details in this biosensor kind of things, like. And mostly what we have obtained is the application, some of the application which is related to my biosensor kind of things, like. So what we did was like a glucose sensor, which we can also blood glucose, which we can obtain those things. So how much glucose content will be there inside that. So this is some of those examples which we have, I'm saying about the by using the biosensor kind of things material, like. And coming to this electrode and all, some of those rapid detection of clinical abnormalities and improve health care treatment and called things like clinical diagnostics and all. Coming to these are all the different synthesizing techniques which I had discussed. And coming to this, like, so after synthesizing, what we can do, what we have to do. So we have to characterize those materials, synthesized material in different characteristics properties. So this characterization, which will be called as an X-ray diffraction, the scanning electron microscopy, even the micro hardness, which we can check, different UV visible, we can measure of those nanometers, nano-sized material, and different kind of drug delivery also, which we can synthesize, analyze through this characterization. And this, some of those phase confirmation and size confirmation, which we can do with the X-ray diffraction, as well as a transmission electron microscopy, as well as the scanning electron microscopy. Even we can see the particle size through the dynamic light scattering technique, like. So, which I will discuss slowly one by one about those things, like. So coming to this, like as we have a crystal size and particle size and grain size and all, like. So we have a little confusion about those students. My students like what is crystalline size, particle size, and grain size. And so you can see here from particle size to grain size, what is grain size to the crystal size in the picture below, which I had shown. Like so, whatever the single particle which we can see in the nanometer range, so that will be called as a crystalline material. Like so, all these crystallite material which will forms come together and which we can observe the crystallographic material kind of things like that. So each grain and all will come together and join together and which can make as a big material, bigger size material kind of things like that. And coming all the grains and all come together, which will be makes as a crystallite size material. All right. So coming to this XRD and all like which we had extensively done with the X-ray diffraction. So what we through this X-ray diffraction, so whatever the material which we have done, whether the material will be in the FCC phase cubic center or BCC or even in the which phase it will be, like all this phase confirmation as well as the material confirmation which can be done through the X-ray diffraction material kind of that. And coming to this, like as we have X-ray diffraction, so which we have done, we will keep those material under the X-rays, and then slowly we can take the reading in the beam of the other side, like collector, like kind of things like. So this, which we had, one of the picture which I had made, which I had done with those things like the face confirmation and kind of things. So like this is the material of X-ray pattern which I had done with the cobalt doped hydroxyapatite material. Like even at figure E and D, which says that like some of those phases of cobalt oxide, so cobalt oxide also is joined together and which will be forming the cobalt oxide dual phase of so biphase, so dual phase or biphase and all which will be absorbed in that material like cobalt doped ceramic material. So coming to the scanning electron microscopy which we have done at Chungnam National University to size confirmation and all like so through the scanning electron microscopy. And this is about the transmission electron microscopy, which is nothing but a little advanced to the scanning electron microscopy. So whereas scanning electron microscopy, which we can observe through 500 nanometer and all those kind of, even for the transmission, which we can observe till up to the 5 nanometer range also through the transmission electron microscopy. So these are all the some of those TEM images of cobalt doped hydroxyapatite which I had said like. So mostly this bioceramic what I'm discussing, this bone material and all, so which is nothing but hydroxyapatite, so which we can say. So calcium phosphate normally it will be having 10 phases. Out of 10 phases, one such phase will be called as an hydroxyapatite, which we can say that. So those hydroxyapatite, which will be having a hexagonal structure kind of things. Like. So this hexagonal structure, which you can see in the TEM image of I as a hexagonal morphology, which we can even in the C also, E also, which we can see the hexagonal structure of a material of hydroxyapatite material. So what initially as we have without cobalt or cobalt incorporation of hydroxyapatite, which will be showing spherical morphology. Slowly incorporating this will be forming as well as rod-like morphology and also the chain like porous kind of material also which we can observe in the very high, very high doped cobalt doped and coming to here, like what I had said, coronavirus structure, like so you can see those 10 nanometer range and all, like the structure of those coronavirus and all, which has been done by the Dr. Atanu Basu, head of the electron microscopy at the National Institute of Virology at Pune. So recently they had published in the Indian Journal of Medical Research. So what they had the structure they need because initially we don't know even still the people are fighting with those COVID. So even that the formation, even the mutation is changing with a different shape and morphology kind of thing. So by making this even a different atmosphere and all, still the studies are we couldn't able to trace out those structure, a proper structure of this COVID virus and all that. So initially, like 4 months back, like this has been published in the Indian Journal of Research, Medical Research kind of thing by the Atanu Basu. So they had distinguished, they had found out about the structure of those coronavirus, which will be in the spherical shape morphology kind of. Coming to the dynamic light scattering and all, this light through the laser light and all, we can detect the size of that nanoparticle kind of things like through the zeta sizer as we have when I was at Anna University and all, we have dynamic light scattering equipment which we can define those size of that material, and also we have we can find out the surface property whether the surface of that nanoparticle is in the nanometers, positive surface as well as the negative surface. So with these uh, we can distinguish so whether the particle is in the positive surface or in the negative surface through the zeta sizer and zeta potential measurement and all. So what these zeta potential and all kind of things like whether mostly this bioceramic which I was discussing, so this will be having surface negativity. And coming to the blood cells and all normal cells which we having in the inside our body, bone cells and all these cells kind of thing will have a different kind of structure like so whether that will be having the positive surface of those things. Due to this positive and negative so combine together so that it will enhance, it will combine together, it can easily cure those cells and all like. So that's what otherwise as we have know that like negative will reflect each other, like as the negative surface will always attract the positive surface which we say that like. So they even the negative particle which always goes towards to the positive surface of the cells and all kind of things like. So even mostly this cancer cells and all, this will be having with the negative surface activity. So that's what we do is we are positive to enhance the increase the my material whatever I said. So bioceramic hydroxyapatite material without doping the metal ion that will be having positive negative surface as I said. When I incorporate these metal ions, which will be having the iron, cobalt, and all, which will be increasing the positivity surface, so that it will go towards the cancer cells and it will be kills those tumor cells and all. Therefore, that will be those cancers and all will be apoptized. Apoptized is nothing but dead. So coming to this vibration VSM sample magnetometer and all kind of things. So what the thing is, whatever I synthesize those magnetic ceramic material, so magnetic material which we will keep in the magnetic property like. So what we had observed is mostly this calcium phosphate. Calcium phosphate is nothing but like similarly as a calcium carbonate like. So calcium carbonate and all usually and they used to keep in the roof and all roof technology and all this ceramic and all material. These will be having a diamagnetic property like kind of thing. When I keep incorporating these metal ions, so what slowly this diamagnetic will be changing to the paramagnetic material. So slow after finally it will reaches the super paramagnetic nature. This super paramagnetic natured material will be used as a targeting those at the particular site and all kind of things like. So, and coming to this, like VSM is one such technique which we can measure the magnetic property of those material. Coming to the hardness and all, which we can say is that hardness, which we can measure, will make the pellet material, and then slowly, which we, by applying the load and all, different load and all, we will be defining those material as a increasing those material into the hardness property kind of things like. UV visible is mostly for the in vitro studies. In vitro means nothing but like a laboratory scale kind of things like. So by making the cells and all human cells, normal cells, cancer cells, so which we can keep in the laboratory. So we will keep these material inside those nanoparticles inside those E. coli bacteria, as well as the materials of cell cultures, and all kind of studies will be called as an in vitro studies kind of. Even blood compatibility, drug delivery, all these in vitro drug delivery, we can say that. So all these studies will be called as an in vitro studies. If we do those things, do these studies, these nanomaterial studies in the in vivo level, in vivo in the sense like as I said, rabbits and mice and all. So there that could be used for those in vivo studies. We can say that. So this UV visible, which could be mostly extensively we can use for those studies of those MTTS, cytotoxicity. So all this toxicity, this UV visible spectroscopy will be very much useful for those studies kind of things. This is about the drug studies which I had says that like even what I did was like I used to do these nanoparticles as a pellet making, and after pellet, so we will keep these pellets, nanoparticles inside the a shaker, orbital shaker, and then slowly we will mix those pellets with the drug also like. So along with the drug, we will keep this material inside the PBS solution, PBS phosphate buffer saline solution like. So up to 50 ml or 100 ml, we'll take those buffer solution. So and then we'll keep these tablets kind of thing. Slowly we'll keep rotating through the orbital shaker kind of things like. And after reaching certain time period, how long this drug has been released, we can observe those kind of things. So after finishing those, the finalization of a drug release, so we can study through these UV visual spectroscopy like kind of thing. So and these will be these drug studies, whatever the drug studies, we have a theoretical models of like a theoretical model like first order and Higuchi and Hickson Cornwell, and some other studies which will be theoretically we will match those whether the Hickson theoretical as well as the experimental will be matching or not. Like we will study those things, and then we'll finalize this will be the better drug release compound material which could be used for the drug release kinetics. And coming to this through the UV, like blood compatibility, as I said, like what is blood compatibility means nothing but like, so even I had sensors with many materials, like thousands of material. So but whether if we keep inside our body, there will be clotting, blood clotting will occur, like kind of things. So through these blood clotting and all, what happen is like, so if it clotting occurs, then there will be no usage of those nanoparticles, nanomaterials kind of thing. So what to study those blood studies and all, we have an study which is nothing but hemolysis assay. So what do is we'll keep these nanomaterials into the human blood or whatever the mice blood or different kind of blood, and so that after several after some time like half an hour or 2 hours kind of thing, so we will take the UV visual spectroscopy reading kind of thing like. So what says these graph says that like the percentage of hemolysis is less than 5%, so then that material will be having the highly hemocompatible material kind. So this will be the standard which will be used for the ISTM standard, like international standards kind of things like. So based on those standards, we can say that this will be having the highly hemocompatible material. All right. And coming to this protein absorption also, which we can do under the UV visual spectroscopy, so which is nothing but like protein and all different kinds of protein which we can keep inside those material, our nanomaterial kind of things. So these are all the different methods, like the explanation of how we can do the protein observe. So we can see here one of those picture, one of my picture which has been observed a more protein of that material. Like so, as I said, like different previously, like which we had doped cobalt atom into the calcium phosphate material. So, as the higher concentration which we of cobalt material, which shows the better absorption of the protein. Like so, this will be protein will have the negative surface as well as the this cobalt material which will be having the positive surface, better, very high positive surface. So due to the surface and negativity and surface positivity of those material, the protein absorption will also highly increasing. So coming to this cell morphology, like so as we have inside our human body, which will be having different kinds of cells, like brain cells, so bone marrow cells, even blood cells, different kinds of cells like so cancer cell, even somebody having the breast cancer, like that will be says as a breast cancer cells, HER2, as I said previously, like so breast cancer cells, and then bone marrow cells, bone cancer cells, and different kinds of cells if we had in that, like so if these one, the cells which I had done with the kidney cells, so kidney cells is which we had taken as a normal cells, like and coming to the cancer cells, which I had taken as an HEK293 is nothing but a kidney cells, human embryonic kidney cells. Like so, whereas coming to the cancer cells, what we do is like we used to cross check with the normal cells as well as the cancer cells. Like whatever I synthesize those material, so that material which we can keep inside the normal cells as well as the cancer cells. So what we do is like similarly, if the normal cells will not die, almost all the cells like so if that material kills whatever my material keep in the cancer cells, if most of the cancer cells has dies means which could be used for the both the cancer cells as well as the normal cells kind of thing. So because why? Because I will do why should we do the normal study as well as the cancer study means like even if we keep inside our body, the normal cell should not die. So that is the main reason for those things. Like if the normal cells will die means like there will be no usage of making those material kind of things. So that's why initially we do with the normal cells like and once if we obtain with the better results of the toxicity range will be very low in that normal cell, then we can keep this material into the cancer cell. So if we obtain this even in the normal cells dies no, very high toxic level, then we cannot proceed with the next further studies kind of things like. So because unless that won't be useful, that won't be usage of the further studies kind of things like. So if it success in the normal cell, then we go with the cancers kind of cell. So these are all the different studies which we can observe through the microscope. So optical microscope which we have, so which we can study those pictures and all. So you can see here HEK as well as the HeLa cells. HeLa is nothing but like a human vaginical cells. So those will be the vaginal cancer kind of things like that will be the cancer which will be mostly occur in the human women's kind of things like. So so this HeLa cells as well as the kidney cells which we have done with the optical microscope pictures and all. And this MTT is nothing but the cytotoxicity which we can do under cell viability. Where viability in the sense like whether the cells has been viable. Viable is living or not like. So cell viability which we can observe those kind of. Coming to the blood compatibility of iron doped nanosize hydroxyapatite is nothing but this is till up to that which I had done extensively whatever the studies which I have been done in my previous research and all those things like there are different studies which can also done like PL studies and for so many other studies which can also be done for other research kind of things. Whatever I did, my research area, which I was focusing on those normal techniques and all characterization techniques. So coming to this, from here onwards, as I explain you some of my research ideas, which I have done in my since from my PhD to till now, like so as I said, like blood compatibility of iron doped nano-sized hydroxyapatite. Like, so iron dope, iron and all will have a very good magnetic property, which I had said. Like so, you can see those magnetic hypothermia and cell separation and all, like hypothermia treatment and all, which can be used. Like so, this we had published in the ACS Material Science and Interfaces, like materials and interfaces, like. So these are all the TEM images like spherical to rod-like morphology. As you can see, those diamagnetic to super paramagnetic nature of the vibrating sample magnetometer. And coming to the drug studies, as we did, as I said, we had extensively done those drug release kind of studies with those nanoparticles. So we have antibiotic as well as the anti-cancer drugs we have taken, like so which is available in the market, like so this anti-cancer drug, which is nothing but the 5-fluorouracil as well as the amoxicillin, is the best antibiotic material, like so this antibiotic which we mix, this amoxicillin mixed with the our synthesized material, and then we will make a pellet, and then we will keep it in the orbital shaker of beaker with a PBS solution. Like so, after several time period, how much the drug has been released from that tablet and all, we can measure through the UV visible spectroscopy kind of. And what we observe is like so very high concentrated iron doped hydroxide ceramic material shows that very fast release and all. So which is having with the for the lower level of iron content, so which shows that a sustained release in that light. And coming to this research, like fabrication of mercaptopurine, 6-mercaptopurine is also used as an blood cancer kind of things, like so this has been used for the blood cancer therapy and all. So what we do is like we did when I was doing my research at South Korea, so which I have extensively worked on the microfluidic channel kind of things, kind of. So this microfluidic channel will keep those hydrogel beads, polymer bead inside those micro channel, and slowly how the drug has been releasing inside that microfluidic channel. So once if we mimic inside those whatever the biosensor kind of things which we can trace out those material, senses those material. So even the same sensor kind of material, if we keep inside that, mimic those on the skin and all. So how the drug has been released to the body and all, which we can study extensively through this studies. And still we have recently sent to the publication kind of things like. So these are all the several applications of which has been done in the microfluidic channel kind of things like. So some of those smart sensing materials which they had used like ocular drug release, like so they even they keep those synthesized material inside the ocular, so eye of the rabbit, which shows that like a drug, how the drug has been releasing inside that, like even the food industry also this microfluidic channel has been used for the even for sensing those glucose inside our body as well as the pH of our normally our pH body temperature body pH will be 7.4 like kind of things like if we excess of those 7.4 and all, so this will be the acidic and basic medium kind of things like inside our body at stomach mostly we will have acidic medium like so that's what whatever we eat, so slowly it will be easily digested. So normally at that stomach area the pH at our stomach area which will be having 5 as an acidic medium kind of things like so even less pH kind of things like. So even if we go with the base media and all, so mostly some places it will be occur in the human body kind of things like. So some of those flexible tactile sensors and even to cure those lesions and all, some of the fluidic channel has been developed by some other people like. So this is my material which I have developed through the microfluidic channel, which we keep those micro hydrogel bead inside the channel and how the drug has been released and all kind of things. So we have done with the photo lithography of those silicon wafer which we've done, and on that silicon wafer which we can keep those PDMS material, polymer material kind of things. So polydimethoxy saline material like. So PDMS is nothing but like. So we will keep those on those polymer on the silicon wafer, and we will make as an a channel kind of things. And again, we will keep as an like another replica of the same material, and slowly we can combine, join together with those things like. So inside that, we will insert a hydrogel bead, and then we will study with the further study kind of things like. So this is the study which we have done at some of the drug molecules which has been done observed on the surface of the hydrogel bead kind of things like. So this is the hydrogel bead which is showing in the B figure B, and those hydrogel bead will cut into two pieces with the help of blade kind of things, and then with those bleed on that bead which we can observe through the scanning electron microscopy. So some of those drug molecules which you can see in the with the higher magnification also like kind of thing. And coming to the drug studies and all which we have done extensively at so done with extensively with the lower pH as well as the higher pH like. So figure A shows that the pH at 5 and blue shows that the pH at 7.4 like. So coming to this after drug, so as we kept the channel as I shown you in the previous slides, so slowly we will send the PBS saline solution through the pedestal motor kind of things like, and after that how the drug has been eroding like kind of things like. Slowly you can see observe those drug molecules at the initial 4 hours how the drug has been eroding like slowly dissolving in the PBS solution kind of things like. So at pH 7.4 and pH 5.5 like. So as we keep on increasing the time interval by using the peristyle pump in the microfluidic channel, slowly the drug molecules of those cubic drug molecules which can be totally disappearing in that like. Coming to this studies, we will inject these cells, human cells, HeLa cells, as well as the kidney cells inside the microfluidic channel. We will send those, pump those cells into the microfluidic channel, and then we will take the pictures of those photographs of those microfluidic channel, how the cells has been growing inside. So what we found is like here the kidney cells, the normal cells is growing very high. It will be there is a vast, wide spreading of those normal cells like. And coming to this bead in the HeLa cells like cancer cell, slowly you can see the thing is like when we go these cells inside for different time interval, initially the cell should be in the round morphology, and slowly after attaching on the surface of the surface, it will be spreading taking place. So slowly the spreads like wings and all, how the wings will be spreading. So slowly these cells is also spreading like that. Like so, what we observe at the HeLa cells, so at particular time interval, again it will be forming a round shape morphology. So which means that which says that the round like morphology which says that the mostly almost all the cells has been apoptized, died. So due to that confirmation, so we can say that these HeLa cells will be dying. and these normal cells will be growing very fast. So like that, we can finalize those materials through the optical microscope. And coming to this rapid removal, like when I do one of my patent, which we had published, like what we done is we have the collaboration with the Annamalai University. So with the Chidambaram, Professor Chidambaram, like what we found is like they visited our lab like at Anna University, and they took our samples, like whether our sample will be the mostly they are working on the water treatment kind of things. Like, so they are from the Department of Geology. Like mostly they do these fluorine content studies and all kind of things like. So what we do is like a biosynthesized material. So this, what they do is they took our samples and then they kept in the, they used to collect the water, groundwater in different places kind of things like. So initially, we too gave many samples to them, and then what they found is, interestingly, the fluorine content has been very good absorbing of my sample, like whatever I synthesized through the microwave as well as the hydrothermal combinatorial technique material like. So after that, what they do is like they just kept this powder material inside the groundwater, and then they used to check with the fluorine electrode like. So they used to check with the fluorine electrode, and then they confirm that okay, this material has been absorbing very good fluorine content like. So initially we too don't know like okay, they said like we mostly for the research kind of things like we used to keep on doing for the several times. Am I right? So based on that, like again, we used to keep synthesizing those for field studies and all. So what they requested us in the Anna University, so they need of 1 kg sample of to test in the field. So for this hydrothermal and microwave, the what will be the yield is maximum it will be like a milligram range. Like so, I used to keep on synthesize these hydrothermal and microwave material up to the half kg to 1 kg, and then we just gave those samples to the Annamalai. And then again, we after repetition of the same material, again and again, we keep finding those material will be good absorbent of the fluorine content. Then we finalize again. We started going with the JNTU collaboration also. So we don't know like whether this still we have the confusion like is it right, we as we are doing right or not like. So we just have the collaboration with the JNTU also New Delhi like. Again they too found the same thing like. So what the thing is like as much as fluorine content is observed even after several usage of the same material like. So that's what we shared here. Absorption, initial absorption till up to 92%, which I had observed. And after regeneration in the sense like we keep those material in the NaOH solution, and then again we started finding the fluorine material, fluorine electrode like. So after the regeneration also, even after regeneration, this material, this fluorine material will formation of the NaF, and so that which will be forming again 60 percentage of after regeneration also. So then we finalized, we started going with the patent of those material, of those synthesized material. So these are all the content of fluorine, which could be like 0.9 to 1.2 concentration milligram per liter, which could be used in the dental caries, like kind of for the children. Even for the dental fluorescence for the elder people, like which shows that 1.5 to 3 milligram per liter concentration, which we can have. For mostly for the 10 milligram per liter, which will be crippling, like as I shown of bending of those skeletons and all legs and all, that could be observed with the 10 milligrams per liter. So this will be one of my paper which we had published in the when I done my postdoc at Politecnico di Torino, Italy. So there we used to do with different kind of materials like so alumina, silica, and zirconia. Even mostly if we having accident and all, they will keep those biometals. So this steel rod and all titanium metal. Titanium is very good biomaterial, like bioimplant material. Like even the niobium. Niobium plays a major. Recently, they started doing research on the niobium material kind of things. So we used to keep doing with a different surfacing of those, changing those surface property of those material and how it cell has been observed. So this human osteoblast is nothing but the bone marrow cells. Like so, after those material like market available material, we just modify those surface modification with the different polymers, and then we have done with the different cell culture studies and all, even the protein absorption studies and all kind of thing. Coming to this, like controlled nanoparameters of those which I done with the chosen university. What we done is like so silver as well as the iron content, combinatorial method of combination of silver and iron. Silver is a very good antibacterial property, as well as the anti iron is having the anti-cancer property. Like so what we observe is like for the silver doped bioceramic, which has been showing a very good, very vast, very rapid release of those drug release content. Like even with the iron content shows that it shows the very wide range of those. So what we observed is very slowly, which has been released those drug molecules. So after mixing this iron as well as the silver, so what we observe is there will be a drug release content in the pulsatile model, like kind of things like a pulse model, which we can observe release those drugs. So we have extensively done our studies with the normal as well as the cancer cells, kind of things like. This is with the drug release kinetics, which we can observe those drug release studies, as well as the blood compatibility studies. And here you can see at the top pictures, so normal cells, mostly you can observe those cells is when spreading, keep on spreading those things like. And whereas in the bottom, figure 2, HeLa cells, when we go with the HeLa cells, it keep on dying. So which says that like as the concentration of those nanoparticle increasing, and slowly that cells will automatically die. So what we do is like we will keep like different 50 milligram, 25 milligram, and something like 100 milligram kind of thing. And then at particular concentration, we'll 25 milligram as a standard concentration, and then we will go with the further cell studies, like kind of things. So this is about the drug study, like irradiation with collaboration with Dr. Ashok and K. Ashok sir. Like recently, we have a collaboration with those scientists. So what we do is by the irradiating our nanoparticles by using the pellet formation, and it will enhances the bioactivity property, kind of things like. So we can study with the different species of E. coli and Staphylococcus kind of things also with the different bacteria. What this has been observed is which cures the bacteria also, kind of thing. And coming to this, which we had published recently in the materials letters, the kind of things like zinc oxide nanoparticles, which can be shown cubical hexagonal structure, kind of thing. So here what we done is like cell viability with cancer as well as the HeLa cells, kind of things. And coming to here, recently we had published another book chapter. What it says that which are different metal dope of all these studies with the different metal nanoparticles. So how the metal doped, so slowly it has been the number of publications which has been increasing, which we have published as one of those book chapter in the Springer, like kind of things like. So here, this is the mechanism which I have discussed. when it was with the external magnetic field and all, these yellow color particles will be the magnetic nanoparticles. So those nanoparticles will stick at that particular site, and it will slowly, it will be and increasing the hypothermia, increasing the what would I say, the heat treatment of those at the external field and things like. So these are all the A and D will show that the cement TEM morphologies of those ceramic material. And then B and E will be discussing about the iron doped hydroxyapatite nanomaterial. And C and F will be the cobalt doped nanomaterial kind of things like. So coming to this, with the during the 1980s, there will be an evolution started taking place of cell separation kind of thing by using these magnetic carbohydrate nanoparticles kind of thing. And slowly after 1994, they started doing with the manganese nanoparticles by the liver imaging kind of thing. They'll image those liver images through this manganese hydroxyapatite kind. And recently at 2009 kind of in 2009, they started doing with the hypothermia treatment by using through the in vivo level range kind of things like. They started in vivo analysis by making iron doped hydroxyapatite. And during 2013 and all, there will be breast cancer treatment for the lamellar. So same similar kind of a scaffold material like they started doing. So even with the gelatin cobalt nano hydroxyapatite, which could be used for the bone cancer kind of things and all, and still the research has to be developed further for the publications of those nanomaterials, magnetic nanomaterials in the cancer treatment. This is about my recent paper, and this is about my some of my publications and patents like. So with this, like, I would like to thank my research guide, Professor Narayan Kalkura, who is the director of Crystal Gold Center, Anna University, and Professor Sylvia, one of my mentor at postdoctoral fellowship at Politecnico di Torino, Italy. Professor Sohn at South Korea, Chosun University. Professor Yang at Chungnam National University, South Korea. And also, as I said, Professor Ashokan sir at IUAC New Delhi, like for their valuable suggestions and cooperation. So without them, like there is nowhere for me. Like so, with this, I would like to thank the organizers as well as the principal and correspondents. So if anyone has any doubts, like please let me know those to clarify your doubts, kind of things. You can unmute or so, Chitra ma'am.
Yes, sir. Yes, sir. We had a great session, sir.
Yeah.
Let me check.
Thank you, sir, for the inspiring session. Sir, your session explored us the application of magnetic nanoparticles for hernia treatment, and very exciting to come across the practical application in human biology. Definitely, your talk will motivate the researchers to innovate and develop further. Now, I request our dear participants to come up with queries for discussion. If you have queries, please raise your hand or else post in the chat box. Sir? Sir, we have a question in the chat box. Shall I read, sir?
Sir?
Yeah, madam. Yeah, coming to the reduction of your organic bulk material to the nanomaterial, as I said, like through ball milling kind of things, we can definitely reduce your bulk material to the nano size material kind. So that's what we can. Many people are using through the. At first, they will anneal these material kind of things, and then after that, they will go with the ball milling kind of things, kind of. So that definitely we can approach, madam. And coming to the next one, like what is hydroxyapatite application? So hydroxyapatite is, as I said, like ceramic material which could be used. So hydroxyapatite, as I said, the calcium phosphate material which will be having 10 different phases, like so as the composition of the ratio of calcium as well as the phosphate. So composition at different composition, if the calcium and Ca by P, which we can say that like, so Ca is nothing but calcium and P is nothing but phosphate. Ca by P, which should be 1.67, the range will be, then that will be called as an hydroxyapatite phase. So, all right. So then it will be having like, it can have even at the 1, Ca by P ratio will be 1, then that will be called as a tricalcium phosphate kind of thing. There will be other phases which will be alpha TCP. Out of 10 phases, I'm saying that alpha TCP, beta TCP, tricalcium phosphate, and amorphous calcium phosphate, hydroxyapatite, all these things like. And coming to the application and all, like, so this will be used very vast application of fluorine removal. So even for the bone replacement, dental replacement, all kind of things which can be used. And how can we measure the toxicity of biosensors? The toxicity of biosensor, which we, as I said, which we can measure through the cytotoxic cell viability and all like. So through the cell viability, what we do is we'll keep this biosensor material inside the Petri plates like. So then we started keeping those Petri plates. So we will keep these Petri plates, we will keep the biosensor inside the Petri plates, and then we'll insert those cells like a normal cells as well as the cancer cells. So once if this biosensor, when it reacts with these cells, normal cells or cancer cells, then we can, the toxicity range, if it kills almost all the normal cells, then there will be high toxicity in that biosensor material. If the cells, normal cells won't die, then we can say that the biosensor will be having the biocompatibility, better biocompatibility. So through that, we can measure the toxicity range through, there will be several toxicity measurements like MTTSA, we can say, cell measurement, like through optical microscope. We can see through optical microscope whether the cells are spreading or not. Philadelphia, we can say that. Philadelphia in the sense like spreading of those cells. So that will be called as Philadelphia. So if those Philadelphia is keep on spreading, slowly is spreading in that. So that will be saying that the toxicity range is very less content. All right, madam. And coming to that, how can we detect those cancer cells? Like, so normally that this imaging kind of things like scanning and all, if we can say that the normally if we have some injury in that like tumor which is having like mostly if they have somebody ate gutka pan and all kind of things like, so they will have the throat cancer kind of things like when smoking and all the kind of things. So what they will do is if the cancer tumor cells is keep on spreading in that, then initially we can say that there will be there will be sensation of paining or something like. Then after that, we will go with the MRI and all those kind of imaging techniques, and then so that easily we can find out those scannings. All right. So in that like that is about to detect those. What is the purpose of using SEM and all? SEM is nothing but like we can find out the size of those nanoparticles, right? So the morphology of those nanoparticles, like. So initially what I do is like I had synthesized material. So whether my material is in which shape, which size and all, I should know that. So otherwise, so everybody will synthesize some material. Yeah, see, this is my nano size. They can easily, they can find out, they can easily say like that. To confirm those confirmation and all, size and morphology confirmation, this scanning electron microscopy has been used. All right, sir. So that is about the coming to the how do we go about controlling the size and shape of nano during synthesis in the. Correct. Very good question, which you had asked, Dr. Sridharan. Like, so what we do is like controlling morphology and all. Initially, what we'll do, like I had synthesized with a different temperature, like at 100 degrees Celsius with under high pressure and high temperature in a closed enclosure, like. So then I will go with the confirmation with the SEM morphology, like. So what we observed is initially I obtained at particular size of 50 nanometer range. Then again, I will go with the 150 degrees Celsius of those temperature and pressure. Obviously, what will happen if I keep on increasing the temperature, automatically the development of pressure will be increasing. So based on that, increasing in the temperature of 150 degrees Celsius. So what this is the simple example which I am saying like. So one such example like. So again, I will go with the same morphology. Then again, whether there will be a reduction or increment in that size of that particle, like. So whatever the synthesized particle, then we can find fine light. Okay, so if I increasing the temperature, then my nanoparticle size will be decreasing or increasing, whatever that. So that we will make the confirmation through those techniques by doing several experiment kind of thing only. And coming to the surfactant, some of the people will be using by using those surfactants like. So if we use those surfactant like in some of the hydrochloride, like as in some of the HCl or some other surfactants. So what these surfactants will do is they can reduce the size also, like. So at different concentration level. So if we add 100 ml, so then the size will be reduction 10 ml. Sorry, not a, so 10 ml. The size of those nanoparticle will be the reducing. So like this, we can make the confirmation of those size and shape. So that is about the shape and size of those molecules. So nanoparticles kind of. All right. If anyone wants to ask, you can unmute and then you can ask you directly. Like, so I had finished in the chat box. Like I had almost cleared in the chat box.
Sir, one of our participants from YouTube has asked, which software do you use to compute? Sir?
Which
software do you use to compute?
Compute of which one? Like either for the drug drug delivery, or which studies which will be like. So as I use like mostly I use Origin for those analysis and all kind of things, like to inserting and all, like for theoretical measurement of drug release and all. Again, what we will do is we will make it in the based on the formula kind of things. We will fit into those Origin software only. Mostly I use Origin only. So nothing other than that. We couldn't go with the other software kind.
Sir, almost we had done with all the queries, it seems, sir.
All right.
Okay.
So
Thank
it was
you, sir.
nice meeting to interacting with as my main motto, which I would like to have a collaboration kind of things with researchers or young researchers or whoever will be the participants or whoever will be. So that will we can strengthen our research, and so that we can improve our quality of our research publication as well as the project research projects and kind of things like. So this would be the better platform, very good platform to have such a kind of atmosphere. I would like to say that. So with this, like if anyone, anybody, any help, like as I have a facility, whatever the facility which I have in and around, so I can do whatever I can help. So that we can interact ourselves, we can enhance our research publications and research activities, like as we did with a wide range of research, like it should not be wasted or whatever the things, as my intention is like that. All right.
I extend my gratefulness on the behalf of our dear participants for our guest of honor for all the inspiring answers. Thank you all for the scintillating participation. I extend my invitation to all the beloved participants to take part in the noon session on energy storage devices, lithium ion battery, and beyond at 1:45 p.m. Feedback link will be provided in the afternoon session. Have a good meal, everyone, and join us. Thank you.
Thank you, sir. And definitely we will have a collaboration with you for our research developmental activities, sir. Thank you. Thank you for your lecture, sir.
Thank you, madam. Thank you. Thank you, one and all. So I'll exit, madam.
Yes, sir. Yes, sir.
Thank you.
I extend my invitation to all the beloved participants to take part in the noon session on energy storage devices, lithium ion battery, and beyond at 1:45 p.m. Thank you all.